Resetting. What are pd1 and ctla4, and what are their functions? Describe two characteristics that pd1 and ctla4 share in maintaining healthy homeostasis, and two that distinguish one from the other. pd1 and ctla4, critical immune checkpoints in health and disease. The human immune system must distinguish between harmful invaders and healthy self-tissues. While activation of immune cells is essential to eliminate pathogens and abnormal cells, including tumor cells, it must be tightly regulated to prevent excessive inflammation and autoimmunity. Two key immune checkpoint molecules involved in this regulation are programmed cell death protein 1, pd1, and cytotoxic T lymphocyte-associated protein 4, ctla4. These inhibitory receptors serve to dampen T-cell responses under physiological conditions and have become pivotal targets in immuno-oncology due to their roles in enabling immune evasion by cancer cells. Functions of pd1 and ctla4. pd1, a member of the CD28 superfamily, is an inhibitory receptor primarily expressed on activated CD4 and CD8 T cells, as well as on B cells and natural killer NK cells in certain conditions. Its ligands, PDL1, B7H1, and PDL2, B7DC, are upregulated on tumor cells, myeloid cells, and other tissue cells in response to inflammatory signals, particularly interferon gamma, IFN. When PD1 engages with its ligands, it recruits phosphatases such as SHP2, which dephosphorylate key signaling molecules in the TCR, T-cell receptor, and CD28 pathways. This leads to reduced T-cell proliferation, diminished cytokine production, e.g., Illinois 2, IFN, impaired cytotoxic function, and ultimately a state of T-cell exhaustion, a hyperresponsive state often seen in chronic infections and cancer. ctla4, on the other hand, is also a member of the CD28 family and is predominantly expressed on regulatory T cells, TREGs, and activated effector T cells. Unlike PD1, which acts at the effector phase, ctla4 functions primarily during the priming phase of T-cell activation in secondary lymphoid organs. It competes with the co-stimulatory molecule CD28 for binding to B7 ligands, CD80-CD86, on antigen-presenting cells, APCs. Because ctla4 has a significantly higher affinity for B7 molecules than CD28, it effectively inhibits the co-stimulatory signals required for full T-cell activation. Moreover, ctla4 on TREGs contributes to immune suppression by removing B7 molecules from APCs via transendocytosis, reducing their ability to activate other T cells. Shared characteristics in maintaining immune homeostasis. Despite their mechanistic and spatial differences, PD1 and ctla4 share two key features in maintaining immune tolerance and homeostasis. Negative regulation of T-cell activity. Both PD1 and ctla4 function as immune checkpoints that inhibit T-cell activation, thereby preventing overactivation of the immune system. This inhibition is critical for maintaining self-tolerance and avoiding tissue damage caused by an uncontrolled immune response. For instance, mice deficient in ctla4 develop lethal lymphoproliferative disorders shortly after birth, highlighting its essential role in immune regulation. Inducible expression on activated T cells. Both receptors are upregulated upon T-cell activation. They are not constitutively expressed on naive T cells, except for ctla4 and Tregs, but rather are induced in response to antigen engagement. This ensures that immune inhibition is tightly coupled to the magnitude and context of the immune response, providing a feedback mechanism to limit inflammation and promote resolution after antigen clearance. Distinct features of pd1 and ctla4. While pd1 and ctla4 share a role in immune inhibition, several important differences distinguish them in terms of timing, location, and mechanism. Site and timing of action. ctla4 acts early during the immune response, mainly within lymphoid tissues where T cells encounter APCs. It regulates the initial activation and expansion of naive T cells. PD1 acts later, primarily at peripheral sites such as inflamed tissues or the tumor microenvironment, where it modulates ongoing immune responses, especially in chronic stimulation scenarios. Mechanism of inhibition. ctla4 primarily interferes with costimulatory signals by outcompeting CD28 for B7 binding and through mechanisms such as ligand depletion and signaling through phosphatases. PD1 directly suppresses intracellular signaling cascades by recruiting SHP2 phosphatases to the immunological synapse, thereby blunting TCR signaling pathways like PI3K-AKT and RAS-MEK-ERK. These differences have therapeutic implications, as they inform how immune checkpoint inhibitors can be strategically applied in cancer treatment. For example, anti-ctla4 antibodies, e.g., ipilimumab, can enhance the priming of T cells, potentially generating a broader and more polyclonal immune response. In contrast, anti-PD1 antibodies, e.g., nivolumab and pembrolizumab, reinvigorate exhausted T cells within tumors, thereby restoring their cytotoxic function. Clinical relevance in cancer and autoimmunity. In the context of cancer, many tumors exploit these checkpoint pathways to evade immune detection. Overexpression of PDL1 on tumor cells, for example, engages PD1 on T cells to induce dysfunction and allow tumor persistence. Similarly, ctla4 activity in the tumor-draining lymph nodes may limit the priming of tumor-specific T cells. Blockade of PD1 or ctla4 using monoclonal antibodies has revolutionized cancer treatment by releasing the brakes on the immune system. These therapies can produce durable responses in a subset of patients, particularly in cancers like melanoma, non-small cell lung cancer, and renal cell carcinoma. However, immune checkpoint blockade also carries risks, notably immune-related adverse events, IRAEs, such as colitis, dermatitis, and endocrinopathies, due to the loss of immune tolerance. Beyond oncology, ctla4 and PD1 are also implicated in autoimmune diseases. For instance, mutations in ctla4 or PD1 genes have been linked to autoimmune lymphoproliferative syndrome and type 1 diabetes, respectively. Therapies that modulate these pathways, such as ctla4 Ig, abatacept, are used to treat autoimmune diseases like rheumatoid arthritis by dampening excessive T-cell activation. Conclusion. pd1 and ctla4 are essential immune checkpoint molecules that maintain the immune system's delicate balance between activation and tolerance. Though both serve as brakes on T-cell responses and are induced upon activation, they differ in their timing, anatomical sites of action, and molecular mechanisms. Understanding these differences has been crucial in the development of checkpoint inhibitors for cancer therapy and in the management of autoimmune and inflammatory diseases. As research continues to unravel the complexity of these pathways, it holds promise for more precise and personalized immunotherapeutic strategies that harness the immune system without compromising safety.